Hello, everyone. Thank you for coming. Before we get started, any of you would like to introduce yourself, maybe through the chat? Like, what brings you here, or where are you coming from? I'm pretty curious. Feel free to, speak in the mic, or you can also type in the chat. I'm currently in the United Kingdom. Irish, where are you currently based at?
I'm currently in Canada.
That's great.
Yeah.
Great, Him. Nice to see you. Awesome. Sakina, rising senior. I'm here to get more knowledge of the research competition. That is cool. Nigeria, that is excellent. Welcome. Hello, Alexis. I'm a student at the University of Waterloo, and I'm very interested in brain and neuroscience in general. Yes, I hope, Arushi will lead the workshop today, and hopefully you'll learn something new about neuroscience. Hello, Herm. I hope I pronounced your name correctly. Welcome. So Herm says, I'm a high school student who is interested in medicine and neuroscience. I'm excited to join the Connectome Research Competition and learn a lot. Well, hopefully you will learn a lot today. Thank you for sharing. Okay, I think we are ready to get started. So again, welcome everybody. I'm Sapo, a PhD student at the University of Oxford. I personally use computational neuroscience to model the brain, but using the human connectome. But as you might see, human and fly, they are sort of similar. They are the brain after all. So I am very excited to be co-chairing the 2025 Fly Connectome Research Competition with Arushi Vardhan, who will be leading the workshop today. So Arushi is the academic ambassador of biological sciences at Clematis. She is also a graduate student in the Department of Cell and Systems Biology at the University of Toronto. Her research focuses on the connectome-driven exploration of neuroconnectivity in the Drosophila visual system. In partnership with Professor Grundmans and Professor Anreider's research group, and on behalf of Clematis, I am very, very excited to welcome you all to take part in our 2025 Fly Connectome Research Competition. But before I pass the baton to Arushi, I wanted to, quickly say the motivation or rather the story behind this competition. So, during our high school and freshman years at the university, we always dreamt of the opportunity to work with the brain data. And I'm very sure that the brain fascinates many of you here. In fact, I think that is why a lot of you are here, is because you like neuroscience, you like to learn more about the brain. Then, came a series of open source, large scale, synapse level resolution brain dataset, or in other words, the connectome. But in particular, the recently released Fly Connectome dataset that we are using for the competition and the tools used to visualize and analyze the Connectomics dataset were packaged in such a way that is very accessible to everyone, regardless of their background and their programming experience. And therefore, we wanted everyone to get a hands-on research experience to explore a real brain dataset using cutting-edge connectomics research tools while being guided by supportive, knowledgeable instructors throughout the research competition. So Arushi and I hope that you will gain a valuable research experience in connectomics, which is, one of the more recent subfields of neuroscience. Connectomics is now playing a crucial role in revolutionizing our understanding of how hundreds of thousands of neurons in our complex brain interact with each other to produce behavior and cognition. And we hope that the competition helps you appreciate the complexity of the brain and also the power of connectomics in neuroscience. Okay, I hope that was a good introduction, and that's it from me. I will let Arushi take it from here.
Thank you so much, Sapo, for the excellent introduction. My name is Arushi. I am a MSc student at the University of Toronto under Professors Gruntman and Anreider. And today, I'll be taking lead over the workshop. And more or less, let's begin. So, a brief workshop plan. So, I will briefly touch on the history of connectomics and the Fly connectome in general, and I will then go into the optic lobe connectome. I'll go into the competition overview and like how to get started, how to participate, and I'll also give a live demo of how to use these tools called neuPrint and Neuroglancer, and then we'll end with an FAQ for questions that have frequently been asked, and also a turn for you to ask questions you might have. Let's first start off with the basic primer on neuroanatomy. Neurons are the basic structural unit of the brain and the nervous system, and basically a cell. And in a sense, how they work is they take in information from other neurons and they transmit that information to other neurons. So how does this anatomically actually work? So a neuron might get information from another neuron through something called dendrites, which take that information. And at the axon hillock, this information is integrated, and then the neuron decides, should I send this information or maybe leave it? And then if it's a go, then that information gets transmitted across the axon to the axon terminal. And at the axon terminal, we have these gaps called synapses. And this is very essential because this is where the communication happens between different neurons. So in this case, this is a chemical synapse, which has a presynaptic cell and a postsynaptic cell. And since it's a chemical synapse, neurotransmitters are released, which that a receptor accepts at the postsynaptic cell, allowing for this communication to occur. Although this is our canonical understanding of how a neuron is structured based on, this is probably a figure you've seen on your textbooks probably, fly neurons are a little bit different, and I'll drive that point using this T4 neuron. Basically, you can see in this image how that dendrite and the axon terminal are quite well separated. But in invertebrates like a fly, you can see that this axon and the dendrites are pretty much mushed together, and postsynaptic, presynaptic are sort of all over the place, and the soma is kind of at the side. So that's just something I just wanted to point out. And also, when you will be exploring neurons using the connectome, you will realize that their structure is slightly different from what we know from high school and university. So going on to neurotransmitters. So as I mentioned, these signals can be transmitted via neurotransmitters, the presynaptic information to the postsynapse, and there are many different types of neurotransmitters in the fly. So excitatory neurotransmitters increase the probability of the next neuron firing, and inhibitory neurons decrease that probability of the next neuron firing, whereas neuromodulators are neurotransmitters that have a broad influence on many different neurons. So an example of an excitatory neuron in the fly is acetylcholine. Inhibitory is GABA and glutamate. And if you work with mammals, you're probably like, oh, that's interesting because glutamate is excitatory, right? But actually in the fly, it mostly tends to be inhibitory. And neuromodulators such as dopamine, serotonin, and octopamine, sorry. And these modulate how neurons respond to signals, often influencing motivation, learning, and behavior. So now let's touch on a brief history behind connectomics. So what is a connectome? A connectome is a basic neural wiring diagram of the brain, or like a brain map. And what I want to drive here is that connectomes are very hard to build. So you see, the first connectome we had, the first few connectomes that we had before the Fly Connectomes came out were of three species, with the most well-known being of C. elegans, which is this worm you can see over here. And the reason this process took like more than a decade was because people literally had to manually like cut layers and layers of this organism and use electron microscopy to image them, as you can see in this image here, and manually trace each neuron layer by layer through these EM data. And as you can imagine, that is an incredible achievement, but it is incredibly time consuming at the same time, and which is why it took so long. So now, because of technological advances in data acquisition techniques and just AI in general, we have a full adult fly brain connectome.
Just to interrupt, C. elegans, the worm, only has, I believe, 300 neurons.
100 neurons, yeah.
And it took
exactly.
so long.
Exactly. Yeah, thank you. Exactly. The C. elegans, this tiny little worm just has 300 neurons, whereas this fly has 140,000 neurons. So we've really scaled up. And this is because of automation of a lot of things, such as tracing neurons and so that sort of thing. Now, there are also more specific connectomes, such as the Fly Optic Lobe connectome. This is the connectome in particular you'll be using. This is from HHMI Janelia, who have released this data set. And the reason the Fly Optic Lobe is such a beautiful thing to study, and is what I studied in my own research as a master's student, is because, well, the fly uses literally most of its brain. Most of its brain is just dedicated to seeing things. And also, there are many similarities between the fly and the mammalian retina. Also, the fly eye is highly stereotyped and crystalline in structure. What do I mean by this? So let's say I find a type of cell connected to another type of cell, and I'm like, oh, that's a very funky connection. Like, that's interesting. I want to know more about it. But the great thing about the fly eye is that there's so many repetitions in this family of cells. So let's say cell A has like 800 repetitions and cell B has 800 repetitions. And I'm curious, is this repetition kind of just a random, is this, sorry, connection a random one or is it genuine? And that's where the fly eye is such a great place to ask these questions. And let's say that interesting connection is repeated 800 times, you're probably like, oh, this is probably doing something really interesting biologically. So that's why it's very important. And very importantly, this fly optic lobe connectome is fully annotated. What do I mean by that? Now, to people like you and I, probably seeing these two neurons, you're like, they kind of look the same to me. I can't say the difference, and that's pretty fair. So the great thing with annotation is that these neurons have been labeled. So I know this one is LC10A and this green one is LC10A, as like LC10B, sorry. And you can see that they're in the same family of LC10s, but they are of different subtypes. And that's really helpful for researchers to use to explore connectomic data. Now I want to touch a bit on fly optic lobe anatomy. And the reason I am doing this is because a lot of the name of the neurons are based on their anatomical location they're found in. And as you will be exploring the connectome, this is helpful to know. And also this is all in the guide and everything. But anyways, just to touch on this. So the fly eye is made of like 800 pixels, you can imagine. Or in more scientific terms, that's called an ommatidia. And each pixel has photoreceptors, a lens, and pigment cells all like mushed into it very compactly. So when light transduces the retina and it enters these four parts of the optic lobe, which is the lamina, the medulla, the lobula, and the lobular plate. And then the information gets compact and gets transmitted to the central brain. So one of the cool things about the eye is that the spatial layout of the image you're seeing is preserved throughout the layers of the optic lobe. This is something called retinotopy. So that's one of the really cool things about the eye. And as you can see in the connectome, which is where I got this image from, where you can visualize the dataset and you'll be using the same one, you can see that these neuropils are very clearly defined. Neuropil meaning these four different layers here, lamina, medulla, lobula, lobular plate. So this is the lamina, this is the lamina, this is the medulla, this is the lobula, and the lobular plate. So as I mentioned, vision is very important to the fly, as more than half of its brain space is just dedicated to that. And you can think of this as it's like the first entry point for complex downstream behaviors. And to give examples of the sort of behaviors that the fly does is like phototaxis. So flies tend to follow light and responds to translating objects, optometer stabilization, navigation, approach avoidance, and visual learning. The reason I'm mentioning these really cool behaviors is that if you notice in the competition, we ask pick a neuron and like figure out what it does in the connectome. That can be a pretty daunting task. There are a lot of neurons. So if you're like, oh, I think it's super cool how courtship occurs in flies and I want to know everything about it, then you can go to the courtship literature. Maybe you'll find a neuron there like, oh, this is really interesting. And you can just go from there and pick a neuron if you're interested. This is just another way. And also like flies do a lot of cool things. If you're wondering, well, what about the human connectome? How are we not studying that? Well, for this competitions for purposes, well, it's because the human connectome uses a different way of imaging. It uses something called diffusion tensor imaging, which is DTI, and it's a type of MRI scan that captures white matter tracts. And white matter tracts are basically a bunch of myelinated axons that connect to different regions of the brain. So this sounds a lot like anatomical connections, right? Structural connectivity. The same thing we're doing with the fly connectome. But unlike the fly connectome, these things are not at a synapse resolutions. I can't really see synapse to synapse which neuron is connecting to which neuron. So, and to illustrate this point, remember 300 neurons C elegans, 140,000 neurons the fly brain. The human brain is what? Billions of neurons. So we still have a long way to go. So now let's enter into the competition overview and what this is all about. Just a brief overview. So the point of this is that you get to explore a real-life neuroscience dataset from the fruit fly optic lobe connectome. This is a dataset I use all the time, every day. This is what my research is based on. You get to use visualizing tools such as Neuroglancer and neuPrint to investigate and visualize these neurons. You get to pick a neuron you like, you study its inputs and outputs, and you suggest how that neuron and its structure might be playing a role in the fly. And there are two-part submissions we have, a 500 to 1,000 word report. There are some guiding prompts we've given in the competition package, which you can look over, and they're by no means that you have to follow them. It's just a guide. And there's also a creative Discord submission. Also, we have $500 in prizes, high school and undergrad categories. So key dates to remember, there's a final deadline, and that is December 1st. And there's an early submission deadline that you can receive feedback from me, which is August 31st, 2025. So the eligibility criteria. So, well, the competition is open to everyone worldwide, provided you're in high school or undergrad. So there are two prize categories. So high school category is defined as students who at the time of submissions are currently enrolled in high school or are on a gap year but have not yet begun post-secondary studies. An undergrad category is defined as for students who, at the current time of submission, are currently enrolled in a post-secondary institution, a college or university, and have not yet graduated. So awards. So there are a total of $500 in prize money, which will be awarded across two categories. So the high school category is the best entry will be a 150, and the two runner-ups will get 50 each, and the same goes for the undergrad. Now, you probably want to know, how do I start? How do I participate? So now I'll briefly go into what the steps would look like to participate, but I'll also be giving a live demo later. So first thing, you go to Neuroglancer. We have provided a link, and I'll show you how you access it. And you choose a neuron from the fruit fly optic lobe connectome. If you want to, just explore, you're free to do so. That sparks your curiosity. Or if you're a bit like overwhelmed, like, whoa, that's too many neurons, I don't know what to do. Maybe there's a circuit or behavior you really find interesting, and you can pick a neuron from there and go from there too.
Yep, just a reminder, Neuroglancer is the visualization tool,
Mhm.
and Arushi will also show a live demonstration of how to use it later on.
Yep. Yeah, exactly. Thank you so much. Yeah. So Neuroglancer is how you actually visualize the connectome data. And so using Neuroglancer, you visualize its structure. So here you'll be seeing like, ooh, like how does this neuron branch? Does it innervate so-and-so neuropils? What visual region is it in? And those sort of things. And then you will take that neuron and you will go to the scientific literature to find out what is currently even known about this neuron. Is it being studied? How is it being studied? Is it known to be a part of a really important visual circuit? What kinds of behaviors might it be playing a role in? Then you'll go to neuPrint. neuPrint is an ecosystem where we store connectome data. Connectome data is massive, so this is where it goes. And here you can explore upstream and downstream connections from your chosen neuron of interest. For instance, I like cell A neuron, let's call it that. Yeah, it might be in the optic lobe, but it might be playing a role in some complex behavior, right? But it's good to understand like how is it connecting to other neurons? How is this chain of information going? So that's what neuroprint will help you figure out. And you can place your neuron in a bigger context of the brain. Then this is time to write. You propose how the structure, every cool thing you learn from the connectome, from the literature, how they might be relating to the structure, the function of the neuron or the behavior that it's a part of. And you will also see whether the 3D structure actually informs any insights this might drive for you. So this is the part where you actually integrate all that knowledge together. And step 6 is your two-part submission. So there's a community submission and Discord. This is your time to be creative. Formatting's up to you. Think of it like a bit of a teaser for what you found. You can make like animations, or if you're into, I don't know, writing poems, you can write a poem for your neuron. I don't know, it's up to you. Be as creative as you want. And more importantly, make sure you post it under the Fly Connectome channel, which is on the Clematis Discord. And if you're not on the Discord, please do sign up for that. There's also a research report submission. You will upload your 500 to 1,000 word report through the competition's Google form. This Google form link will be found in the handbook. Also, there's a report template, once again, just to remind. So if you're a bit lost, how do I even write my report? There are some guiding principles over there. Also, you're welcome to submit multiple entries, but each should follow that same two submission format. But they should also focus on a different neuron to be counted as a new entry. So now let's go into selection criteria and how the submissions will be evaluated. So we will be evaluating both the Discord submission and also the written report. Firstly, we'll be looking into the biological insight for the set submission. So, does it draw meaningful connections between the neuronal structure and the possible function or behavior this neuron is lived through? And have you used the literature and the observations from the connectome very effectively? Does it show curiosity and the ability to explore how the neuron fits into a broader brain system using evidence and reasoning from multiple resources, papers, stuff you've even learned in high school, and the connectome itself? And it shows a real depth and a real understanding of what this neuron is doing. Also, the second criteria will be engagement with connectomic tools. Were the tools used thoughtfully and effectively, Neuroglancer, neuPrint, to investigate the structure, the connectivity, and how were those insights used and integrated into the Discord submission and the report. Screenshots, how the visual representation of the neuron to meaningfully highlight your findings, and just an appreciation for how large-scale connectomics is, well, adding a lot to neurobiological insights. Also, another one would be focused on communication and clarity. Is the written report clear, easy to read, and the ideas are very effectively communicated? Is the Discord presentation engaging? Is it accessible, appropriate for a peer student audience? Also, scientific terminology jargon is defined appropriately with clear explanations. And fourth is learning journey and reflective thinking. It shows evidence of genuine joy in learning and exploration, reflects on the challenges, surprises, or the new understanding and, I guess, evolution you went through of before the connective and after learning about the connective, if that's the case for you, and makes it very clear how this thinking has evolved. So now let's go into how to use neuPrint and Neuroglancer. So firstly, here's the package of resources that we have provided. And if you joined this competition through LinkedIn, the Discord, or the Instagram, we have this link to this package there. We'll also show a QR code soon, but this is all you need. Sapri, you want to say something?
Yep. So I have put the link to competition
Okay.
material is tinyurl.com/flycomp 2025. So that should take you to this Google Drive. Yeah.
Okay, fantastic. Thank you. Yeah. So that's where all the application materials are super important. This is where everything is. So the first thing is the research competition handbook. A lot of things I'm saying is already in the handbook out here. It gives you details on the competition and the submission criteria and all that sort of stuff. The second is exploring the fly brain, a starter guide. This is anatomy and all sorts of stuff over here. So if you forget something and stuff, you can always revisit this. And it also has demos on how to use the Neuroglancer and neuPrint. And three is the research report template, which you can use to guide your report. But for now, we're looking into Neuroglancer. So let's scroll down. Okay, so Neuroglancer, you click on this link over here. So under Neuroglancer, and it directs you to that directly. No sign up, nothing required. And it should take you to the Optic Lobe version 1.1 dataset. So over here. And you're free to also follow me through when I'm demonstrating. So, you remember what's going on. Again, the demo and everything's on the guidebook.
And it's recorded as well, this session. So it will be
Yeah,
uploaded on our YouTube.
yeah, exactly. So let's say, okay, where should we do? All right. So these eyes, well, are to see stuff. So the neural shell, if you click on it, like you'll see the shell. And to actually rotate the structure, if you just click on your mouse, I'm using a Mac, you can just like rotate it. So yeah, it's going crazy here. To zoom in and out, you use your two fingers and you can zoom in, zoom out. And if you want to spatially like actually move the thing, you click shift and click on your mouse pad and you can actually move the brain like that. So let's go into ROI. So if I click on ROI, this will show me the region of interest. And this will show me the different neurobuilds of the optic lobe. So you see, this is the lamina, the medulla, the lobula, and the lobular plate, clearly defined here. Okay, so now let's pick a neuron. So my favorite neuron is CD1 because it looks really cool. So here, okay. And because the ROI is kind of messing with us. Okay, as you can see, it's massive and it's kind of going everywhere, doing its thing. So I can move it around, I can see it, cool. And maybe you're like, okay, I want to know which layers of the optic lobe it's actually innervating. and this is what you would do. So this is where neuroanatomy is helpful, and you might want to have that guide open. But first, let's get this straight. Yeah. So I can tell like there are two places this is innervating. So this is what? The medulla, and this is the lobula. So okay, let's go to the medulla then. So I'll go to medulla, which is ME, and go to layers because I want to see which layer is innervating. So I'll open the eye and I'll click it here, and let's open all the eye. So yeah, all the layers. So now one by one, I can see, well, it's kind of at the bottom. So, well, it's because I know CT1 well. I know it's innervating layer 10, but you probably have to like play around and figure out which layer is innervating. So this is layer 10. As you can see, it's innervating. Go the medulla and same stuff. You can see the other part of CT1 is going through the lobula. So let's find LO. LOP is lobular plate, which is this. So LO. LO layers. Click on this. And now you can see it's innervating layer 1. One by one, you like remove the eye to see which layer. So now I know, okay, CT1 is going to 10th layer of the medulla and the first layer of the lobula. And yeah, so this is an example demonstration of CT1. But let's say um, you want to play with another neuron. Or you don't really have a neuron of interest and you just want to go random. So you can just scroll and, just pick a random one. Let's go with,
Arushi,
yeah.
I have a question. Which is layer 1 or layer 2? It is really confusing.
Thank you for asking. I'm really glad you must drill with me questions. This is the time and place. Okay. Layer 1, layer 2 of like I'll go with the middle. It's a bit easier to see. So let's go back here. So these are all the layers, okay, of the medulla. And it's not transparent, so it kind of hides your neurons, so that's the sad part, but it's fine. We'll work with it. So this top is layer 1. This is layer 2, layer 3, 4, 5, 6, 7, 8, 9, 10. Make sense? So yeah. So if you see when I'm taking off the layers, they're actually numbered as well. So you can tell, oh, that's actually layer 1 or that's layer 4. So like that.
There's another question. So for novice, how will you know you are in the medulla or lobula?
Yeah. So you need a bit of training with the eye. Okay. So how I would go about it is let me get rid of CT1. It's a bit distracting right now.
Another
Yeah.
question is, what do the different layers signify? I think all of these questions are the same.
It's a bit hard. When I started using this, I know it's really confusing and don't worry. And I'm really glad you're asking things. But you kind of sort of memorize the structure and it gets easier over time, trust me. So for instance, if you go to the handbook, so the layers are the lamina, the medulla, the lobule, and the lobular plate, right? So here you have it goes down sequentially. So this is the lamina. It's incomplete in the optic lobe connectome. This is the medulla, the big part over here. The lobula is this part, and the lobular plate is slightly angled off. So that's how it is. And the layers, what do they signify? So the layers, what they signify, maybe I should explain how the connectomes are built and how they define things like neurons. What is a neuron type and stuff like that. You don't just get a handbook from nature saying like that is T5, that is M. No, it's a mess. And this a literal mess. There's no better way to explain it. So this is how neuroanatomists, and this is going back to Cahal, the great neuroanatomist, it started from there. This is how they define cell types is or neuron types is where it connects. Who does it connect to? Is it repeating? That's a cell type, neuron type. So that is the significance of the layers. And it's just to get an appreciation for how connectomes are built, how neurons are made, and how this beautiful structure is structured inside this well. It looks a bit tiny, but it has 50,000 neurons in here. Yeah. I hope that answers your questions.
Sorry, can I
Yeah,
interrupt?
yeah, go ahead.
So to answer Celestine's question,
Hmm.
I think what we should do, Arushi, is we should screenshot this and then we will label.
Okay.
And then we will either post
Yeah,
it on social media and I hopefully it will help you guys. And then you can then now on your own rotate around and try to guess which is, or you can actually screenshot right now as well. And
yeah, absolutely. I'm happy to do that.
yeah,
I think that's a great idea. That would help for sure.
we'll do that after the talk. Great,
Yeah.
go on.
Thank you so much. Yeah. So that's how it works. Where was I? I was going to show another neuron. Okay. Uh, Don't be scared. You'll get used to it. It's confusing, but you'll get there. You'll be a pro. Okay. So let's see. I want to find a cool neuron. No, let's go. Okay. Okay. I honestly have never checked out T2. So let's go there. So you can see this one's looking a lot different than CT1. And you can see it's innervating the medulla and the lobular plate, like it's going, and it's going at like the edge of the lobular plate. And this is the soma, and these are the dendrites and the axons here. So this is just an example of another neuron. And oh, also one more thing you might be confusing you. What are these numbers? 87112, 87495. And they're all T2. Remember I said the fly eye is highly repetitive. There are like several repetitions for a type of neuron you're interested in. So it doesn't really matter which uh, neuron you pick, provided it's the same like type, like T2 is the same type. It doesn't matter what number they are. It's just a repetition throughout the eye, repetition, repetition. So like this is also T2, but like you see, it's a repetition. It's the same thing. So yeah, you can observe its structure and see like where it's innervating. So that's just another example. This is not required, but if you're interested, you can see the pre-synapses and the post-synapses on these neurons. So like that looks pretty cool. And maybe like you should show CT1s. Like it's a lot. Like you can see, like look at this one. This makes like it's connecting everywhere, literally at this point. So yeah. Yeah. So that's something else you can do. But yeah, that's a Neuroglancer tutorial. I hope things are clear. If things are confused, that's great. Point of the competition is also to get to learn these things.
Okay, two questions, and I think we have
Okay,
time, so absolutely go ahead with questions. Is there a reason why CT1 was grouped together and T2 is separated individually? I think that's a great question.
that's an excellent question. CT1 isn't grouped. This is one neuron.
Hmm.
Can you believe it? This mass, and it's an inhibitory neuron. So it's definitely playing some modulatory role, but many, I'm not gonna say it, what it's playing with. You can figure it out. But it plays a very important role in the fly eye. So it's actually one neuron. T2 has several repetitions throughout the optic lobe. So yeah, that's why.
Oh, sorry, we have another question. Can the repetitive neuron innervate different section of the optic lobe?
So you mean one cell type like T2, but like different T2s innervating different
Yes, I
section?
think the question means like can a one neuron from one region innervate a different region in the optic lobe?
To my understanding, no, because that would be a different neuron, right? The classification would be different. Remember, these neurons are grouped by similarity in how they are ordered. So my understanding would be they would be very similarly placed.
But does one neuron cut through different sections? It probably does, right?
Or different neuropils, like
Yes,
different parts.
yes.
Yeah, absolutely. It
Yeah,
can do that. Absolutely.
great. Okay, questions are welcome, so feel free to type in the chat. It's very helpful.
Yeah, that's very helpful for me to know as well. Yeah. And if you have more, feel free to contact us on Discord. Throw in your question, we'll answer it. Now let's go to neuPrint. So remember, this is the ecosystem where you have the connectome data. And this is actually what I use in my own project, except I actually use Python to code with the data itself. But we'll be using the web-based version. So firstly, you need a Google account. So I already have one, so I'm signed in. And you should get access to the Connectome. I have heard instances where it takes a day or two, but you all should get an access. And if you don't, contact us. Make sure you're on the right data set. I don't know why it shows no options. There will be more options. There are a lot of connectomes up here, but for now it doesn't show them. And let's pick a neuron. So this is how you query a neuron or search for it. Let's go for CT1. Oops, C, and yeah, there you go. You put submit. So you'll get something that looks like this. And essentially, this is the ID. Remember, there's only like one CT1. And it gives you the inputs CT1 receives and the outputs it gives. And also, it gives a breakdown of the brain regions it is. And if you're confused what brain regions stand for, you can always like click this and it shows you here. Like this is what they stand for. So yeah. And it also says the consensus neurotransmitter. Um, consensus neurotransmitter because they classified using AI based on a training data set. But anyways, that's not the point of this. Uh, anyways, so yeah, so this is brain region breakdown. So I can tell CT1 receives a lot of its input, like 62% of it in the lobula and 37% of it in the medulla. And the outputs it gives 64% of it go to the lobula and the 34% over the medulla. So this is a bit confusing, this title, but let me clarify that. So inputs hashtag post basically means is the input CT1 is receiving. So if CT1 is receiving an input, it would be the post cell because it's receiving one from the pre-cell. Output hashtag pre is because when CT1 gives out outputs, it acts like the pre-cell and the output is the post cell. So that's what that means. You can click on these individually and you'll find a list of neurons it's connecting to once this loads. Yeah, so like that. But for the purposes of the competition, you can click on this synapse connectivity thing. And it gives you a really nice compact summary of a few cell types that connect upstream and downstream. So you can tell TM9, like it's bigger in size, shows that a lot of TM9's neurons um, connect to CT1 and they connect to the lobular region for that. And also one of CT1's biggest outputs are T5B, T5A, it seems. So you can look here for the synapse connectivity part. Yeah.
Sorry to interrupt. So we have a great question, which is these abbreviations, how would you know what it stands for?
Which ones?
For
Oh,
instance, LOME.
or like I maybe you mean T4A, TM1, T5A, those kind of things. Okay. LO and ME, these are brain regions. You can always get them here as well. These are the neuropils, medulla, lobula, lobula plate. Um, for the connectivity-based ones, like CT1 and those kind of abbreviations, some of them are named based on which neuropil they are. So TM stands for transmedullary. So it's in the medulla, but it's like it's going trans to the medulla. So they are named like that. But some of them are just not. What you can do is for the purposes of finding connections, these abbreviations are actually how most people study these neurons, and it's good enough for the purposes. And if those neurons don't really have a definition, chances are no one really uses them in the literature, so you should be fine. Yeah. I hope that answers your question.
Yep. And just to add also, sometimes you can also just type the neuron name like TM, whatever, and then look at on Google, and there will definitely be some papers on it. And I think that's the point, is to like sort of look around what each of these
And
neurons.
make sure you write fly. I mean, it sounds obvious, but sometimes write fly because these are specific to the fly eye. Yeah. And
Great,
so you
thank
don't
you
get some
for
others.
that answer. We have another question. Each type of nerve has a specific function in the fly, but can I identify its counterpart in the human body? And how can I determine whether its counterpart has the same functions or not?
Hmm.
I can take that question if you want.
Mm-hmm.
Okay. So I'll tell you something about rats first, okay? So we have sort of different animal models to study neuroscience. And why I'm talking about rats is because rats' brain is often closely compared with the humans, because a lot of the brain regions are evolutionarily conserved. So things like, I don't know, hippocampus, which is for memory. It's there in the rats and it's also there in the humans. Maybe there's some source of difference. But let's say with rats, the way we would look at the comparison or the counterpart in the human brain is you'll have to literally look at a neuroanatomy paper. And there would definitely be a review that talks about, okay, this ventral hippocampus is the same as XYZ hippocampus in rats and humans and so on. Okay, so that's with rats and humans. The fly brain is also evolutionary conserved, but a lot of regions might be a bit harder to map to humans. But I think the approach I would take is I would look in terms of behavior. So a lot of behaviors in the fly are really similar to the humans. Of course, the neurons, fly brain, it doesn't have many of the regions that humans have. But for instance, a very basic brain areas related to fly, like especially with the optic lobe, like the vision, it will sort of perform really similar functions that is evolutionary conserved in rats and humans as well. And I think that's the approach I would take. So to summarize, rather than, seeing this neuron is the same as the human neuron, it would not work because fly and humans are just fundamentally really different. But the behavior is really conserved. And so that's why studying the fly is still really helpful in, understanding the neurobiological process of some of these behavior. So I hope that answer your question. If not, feel free to type in the chat again.
Also, to add on to that, there are some like similar. Is it I don't know my mammalian anatomy the best, but it's the ganglion cells, is it from the
Yes.
eye? Yeah, I believe a counterpart to this is like the optic glomeruli. That is not exactly in the optic lobe, it's going more towards the brain. So there are some similarities here and there, but that's rats.
Yep. And again, you just have to, there's way too many neurons in your, even in the fly brain. If you're really interested in specific neurons, you'll often have to search the literature. And I think that's the fun part of the competition.
Mhm.
Cool. Okay, I think we have answered all the questions in the chat for now, but feel free to
All
keep
good?
those questions coming. Great. I'm
Amazing.
glad that was helpful. Okay.
Okay, fantastic. All right. Okay, so all good. So let's go on. Yeah, so now that basically that's the gist of the competition and all you need to know. If there are things that sort of were like, whoa, that was a lot of information went over my head, everything is in the handbook, the starter guide, and the research report template. And you can get that Google Drive stuff, like all these manuals and the handbook up here. You just have to scan this QR code.
Oh, the QR code is on the slide. We have a few more questions. Which database or website are you, I guess, referring to find out more? The dataset that you'll be using are the dataset that Arushi has shown. In terms of websites to find out more, I will compile a link. But if you essentially go to that Google Drive, yeah, it's basically this Google Drive. Do you have any other resources outside of the guide provided that you would suggest to high school students who have little to no knowledge of connectomic and neuroscience in general? That's a great question. We hope that the starter's guide to FlyEM Connectomics, if you read, we hope that we design it in such a way that regardless of your knowledge level in neuroscience, you should be able to actually explore neuPrint and Neuroglancer and
Neuroglancer.
also understand the fly brain. But of course, if you have any more questions, you are always welcome to reach out through Discord. And furthermore, this is not the only workshop we'll be hosting. We'll be hosting more workshops, which we encourage you attend. And of course, all of that will be recorded as well. And the idea of the competition is exactly that. If you have no previous experience with neuroscience, you are able to actually learn something from the competition with a chance of also, being in a community with other participants who are also in the competition. The starter guide's a bit advanced, to be honest. Thank you for letting us know. So we'll have a survey. If you could let us know also, like what you would like to see in our workshops or the starter guide, that would be really helpful.
Yeah, it will help us make sure to provide information that might be more useful as well.
Could we explore other research papers for more insights on the neurons?
Which neuron would you like to explore and why? Haha. Hmm. Do you have a neuron you like? Something.
Oh, I'm not understanding the question really well, but do you mean like,
Him's question.
no, I'm talking about Sakina's question. So could we explore other research papers for more insight on the neurons?
Oh,
Yes, of course. You can explore anything in the literature based on neurons of interest.
and remember, goal of this competition is that you really go out there and like read a lot of papers to get the insight on the neurons. So yeah, absolutely do that.
If you were to do this competition, which neuron would you pick to explore and why?
Do you have one?
Well, I like the CT one. I think that looks pretty cool.
Okay, you pick the CT
Um,
one. I I would pick something in the courtship circuits. I find courtship really interesting. So yeah, something from there. So the LC10s and all are there. Yeah.
yep, that's great. Well, I hope that answers your question. Cool. And also, I'm Celestina. We try our best to make our guide, accessible, but also we hope that this workshop also, help make things a bit easier for
hmm
folks to understand.
Okay, how to stay connected, basically reiterating the points Sapo said. So, resources or things you think might be useful or like future announcements, join our mailing list so you're up to date. Also join the Clematis Discord because that's where we put regular announcements and stuff like that.
And just to add on, this workshop
Oh,
is meant
sorry.
to give an overview of everything, but we'll be hosting more workshops that could even go to the like very fundamentals of neuroscience. And the idea is hopefully we will send you a survey and you could let us know what you would like us to host the workshop on, and we will try our best to actually host those workshops for you guys. Remember, this competition is for you to gain experience, and we would like you to enjoy and have fun and also learn something along the way.
So there are two more questions.
Could you demonstrate how you navigate around the eye? Like, what did you click
Uh,
to see the
so
different segment?
I could, we can go through the presentation and then at the end, I can take up that question and show it again.
That's great. Lovely.
Just remind me. And guidelines for Discord submission. Nope, it's all about creativity. It's totally up to you.
Yes, but I think we have written some ideas on the competition
hmm
handbook. I suggest you look at it, but the gist of it is that you can do anything. It can be a video and so on. Can you make WhatsApp group to us to ask and discuss on it? So the reason why we use Discord is because it allows everyone to ask questions in a systematic way. So like there's all sorts of channel. So that is why we use Discord rather than WhatsApp. So if you don't feel very comfortable asking the questions on Discord, perhaps you can email us. But what we'll do is we'll post the questions on the Discord just for everyone to see. But your name will remain anonymous. I hope that answers your question. Concerning the EM, how are we to incorporate it into the report?
Um, a screenshot, literally. Just take a screenshot or an image of that neuron that you're interested in, yeah,
Yep.
of the data you're looking at. Okay. Um, that's it for me. I will hand it to Sappo after this. But yeah, thank you for listening. I also want to give a shout out to people who made this possible. So thanks to Clematis, the co-chair and program lead Sappo, who's moderating right now, him, our program communications lead, the Clematis outreach team, Ali, Manya, Aditya, and Sahari, and the Grunpan and Anwriter Labs for their support. Yeah.
Great, thank you. Okay, so that is the anonymous survey I have talked about. Right.
Yeah, it would be really helpful if you can give feedback because I mean, this is what I thought would be useful for high schoolers and undergrads to know how to play with Connectome data and get their, hands wet onto it. But if you felt like there were things that weren't explained too well or you want to know more about, please let us know because that is the way I would get to know how to improve.
Thank you. So there's the link. The remaining part of the presentation is just sort of FAQ that has come up. But if you could take the time meanwhile, just to fill out an anonymous survey, remember we will not know your identity. So please give us honest and maybe brutal feedback and any suggestions how to improve. So that's the link to our survey. So that's great. So moving on, FAQs. So can we work in teams? So this competition is for individual participants, and the idea is that each person will submit their own original work. However, we absolutely encourage discussions and brainstorming with other people or with your peers. Just remember that your final submission should reflect your own thinking and individual efforts. We also had some questions around eligibility criteria and graduation. If you complete the submission before you start your university studies, you will be considered under the high school category. And of course, you shouldn't have studied an undergraduate degree before. The timestamp is the time in which you submit. If you have recently completed your undergraduate course requirement, you will still be eligible to compete under undergraduate category as long as your undergraduate degree has not yet been officially conferred at the time you submit your entry, and you have not started a new degree program, such as, a master's or a PhD or a second undergraduate degree at the time of submission. So let's say if your undergraduate degree is conferred on July 1st, make sure you submit your entry before your graduation date, which is any time before July 1st, in order to be eligible. And there was a question about whether would you receive certificate of participation. Yes, all participants who submit their work for the competition will receive a certificate of participation. And the award recipients will also receive the monetary prize and also a certificate of award and recognition. That is it from me. And let's see if we have any more questions. Could you outline what the future workshops be on so the participant can anticipate to join them? Yeah, that's a great question. So I think definitely based on the feedback that we have received, I think we will be doing some workshops just on the actual tools itself and maybe even about the Fly neuroscience, like the fundamentals of Fly neuroscience and connectomics. And we may also be talking about some research papers and give some tips on that. I think that's so far what we have in mind, but the idea is we really want feedback from the community so we can host the workshop that is right for, participants. The idea is so that we all learn together and learn something new about connectomics and neuroscience.
Yeah, and also whenever we have a workshop in it, if you are on our mailing list or you're on Discord, you'll definitely get an announcement for us, so you'll be up to date. So I will go revisit that question. Was it by Celestine? I will again demonstrate was neuPrint, sorry, Neuroglancer, I believe. And so once again, let's go to Neuroglancer. So there's the link. So the question was, can you demonstrate how you navigate around the like what do you click to see the different segments? Sure. Okay. Cool. Okay. All right. So now this is the fly brain. This is the entire brain. This is the eye. And right now the connectome data, we're playing with the optic lobe connectome. We only have the right side of the male eye. So that would be here. So once you click ROI, you will see these neuropils or the different parts of the optic lobe. So, and someone made a really good suggestion. We will label this so it's easier for you to know and orient yourself. But this is the lamina at the top. It's incomplete. This is the medulla. This is the lobula, and this is the lobula plate over here. Okay. So to see that, I basically clicked on this eye that says ROI. And also, don't be scared to click random things. You're not going to crash the server. You're not going to ruin Janelia's database. It's all good. So you can learn, click around, see what things do. It's totally fine. So if in case you forget, don't be scared. But this is how you go. You click on ROI, and that's how you get these layers. Now, to pick a neuron of interest, let's click on C2. So you see that I just clicked on that and now I can see it. But sometimes these ROI, these shells can make it a bit unclear to see. So I can just close the ROI and I can just like see it clearly how it looks to get a nice image. Now to see which layers of the neuropils they innervate, this is where you need to know a bit of neuroanatomy. So this is the medulla, this big part here. So remember, it goes lobula, medulla, lobula plate, lobula. Lobula plate literally looks like a plate. It's like a diagonal plate. And a lobula is this big chunk over here. And you see, then it goes to the central brain over here. So that's also how you can orient yourself. So yeah, so this is where we are. Then let's say, okay, I'm saying this neuron's kind of going all over the place, but it's mostly in the medulla. And it seems to be innervating all the layers of the medulla, as you can see, right? But to really see that, let's click on the medulla, sorry, the where it says layers. this thing here. Then you click on this thingy here where it says hide layer side panel. And you click on the eye, and you should get all the layers of the medulla. And then so like you see, the neurons kind of blocked. You can one by one move each layer and see if you can see the neuron again. So like that definitely innervates layer 1, or that definitely innervates layer 2, layer 3, layer 4, layer 5, layer 6, layer 7, layer 8, layer 9, layer 10. So yeah, that's one way to look at it. So yeah, I hope, Celestine, that was helpful. So there's another question. Oh, what browser are you using? Are you using Chrome, Firefox? Chrome, and it's slow. It does take a bit of time because it's a really big data set to load. I also experience problems. You might just have to wait a bit longer for it to load.
Yes.
Yeah,
Unfortunately,
yeah,
yeah, sometimes, or actually, even depending on the types of neurons, you'll have to be patient. Like in the guide, actually, there's an instruction on how to load actually the entire optic lobe. And sometimes you also need to wait for 3, 4 minutes, because remember, these dataset are large scale dataset. So naturally, it will take some time. And
yeah.
unfortunately, you'll have to be a bit patient with it.
And by the way, you don't have to load the entire optic load data set. That is
Exactly.
not required for the task, so don't worry about it. You just literally have to just visualize one neuron. So yeah, that's all. Someone had a question. I want to describe this neuron. What will I say? So here's the thing, right? You will say, what neuropil is this innervating? Like, where is it going through? What layers is it going through? How does it look like to you? Where are its dendrites and axons? So these are things I can't really just give an answer. It's more like you will get better at this the more, I guess, effort you put into trying to see the morphology and also seeing the literature, how they describe these things. Maybe don't try CT1 because it's pretty big.
Okay, so I think Celestine. So there are a few ways to minimize the data usage, but I think this is more related to the web browser. So for instance, I believe Chrome has some sort of data saver mode or function. Maybe try that. And yeah, that is really frustrating. I'm really sorry.
Yeah.
Have you tried with other neuron, like the smaller neurons? Like
Yeah.
try C2 maybe.
Yeah.
That is a smaller neuron. And I think when you load the neurons, maybe don't click the ROI because now more things are loading. So
Mm.
like switch off the ROI and then click one of the C2 neurons the last time. And remember, Celestine, I know it's a bit frustrating that things take time, but for the report, you just need one screenshot. And we will not judge on the quality of screenshot. We just want to see that you try and understand the structure really well. So as long as you get it to work at least one time, and you put it on the research report, that is great enough. So we will not judge on the quality of screenshots or whatever, but more importantly is how you try to understand these structures, how you create biological insights, and perhaps like link to sort of functions and based on the selection criteria. Yeah, don't worry. And if the problem persists, please reach out to us and
Hmm.
we'll try our best to help. Okay?
And also, if it like helps, remember this competition due date's like December, or the feedback date is August. So there is a lot of time. So don't worry about it. Yeah, I'm
Exactly.
sure it'll work out.
Exactly. Great, but thank you for bringing this up, Celestine.
Yeah,
Appreciate
thank
it.
you so much. There was a question on, can I describe it in the way that I see it, or I must go to the literature to describe it? You should describe it the way you see it. So so remember the point I wanted to bring to the competition is this is the sort of science that really works on looking at things and seeing things. So look at it. And you know, remember, if you're a bit worried, I don't know if I described it in an interesting way, you should just try writing it. Look at the literature and you get an intuition for how people write things. And if you really want feedback, there's always that August 31st deadline where you can send it to us and I can have a look over it and I can give feedback as well.
Yeah. So just to clarify the feedback, you just submit as you would. and we'll give feedback. And
Yeah,
that's about it. And you can
and
choose to make changes or you can make that your final submission. It really does not matter. And to add on Arushi's point, yeah, you can describe the way you see it. In fact, that is the point of science, is like you observe something and then you try to make critical insights. But of course, it really helps. To become a good scientist, you do need to explore how other neuroscientists have been observing these neurons. So you get that intuition and then you describe it and hopefully in your own way, right? And I think that's where you learn from the literature and like search. It's interesting. Yes.
but sorry, just to sidetrack a bit, to explain how do I describe it. So this is what I was explaining, right? One of a very like easy way that doesn't seem a bit like I don't know how to describe this is just to see like where is it going. So for instance, this is the medulla, remember? Like this neuron's literally covering the whole of the medulla. So that is some way I would describe it. Or I could say like its cell body, which is remember the fly cell body is like kind of left at the side, is sort of like here. It's near the lobular plate. You could say something like that. So that you are describing where it's going, how it's traveling, which neuroperson. So that's way to describe it too. Or even goes up to the lamina by the looks of it here. So yeah. But obviously you can't see the complete thing because the lamina is not full.
Okay. I will wait for more people if they have any questions. But I have a question for you. Why is it important to build a connectome in the first place?
Great question. You might, from the sounds of it, saying like that looks like a lot of work and that looks hard. And a lot of people say that. So you're not the only one. Why build these connectomes? Well, one of the hypotheses in neuroscience in general is that people think structure relates to function. And we have found there are many beautiful papers out there in the fly literature where you clearly see an example of that. And just going back to humans as well, people think if we have a complete neuronal wiring diagram of, let's say, a human, maybe we'll be able to understand what goes wrong in someone's wiring diagram if there's a neurological disease or there are mental health disorders and that sort of a thing. And another analogy I really like to think about is like imagine you're exploring a new city and you're flying using a plane and you look down. You can kind of see like, oh, I see like the ocean there. I see an island there. I see a shop there. I might check it out later. I see something cool there, but you don't really know it. Your understanding of the city is fogged. But when you have Google Maps, when you walk those streets and you're like, ooh, there was a shortcut that goes to that shop there. That is the way to go. And that foggy vision didn't really show me that. That's kind of what a neuronal wiring diagram can help us. If we know connections at the synapse level, that is a solid proof, a solid way of saying these two neurons are connected, anatomically at least, not necessarily functionally, but they're connected. We can then go and ask questions or generate hypotheses. If these two neurons are connected, what do you think they might be doing? And then you can test those things out. So that's why building Connectome, albeit a pretty daunting task and one that requires remarkable perseverance to get through, might really be worth it.
We have another question. I'm asking for someone interested in medicine. How would this combination benefit someone who might be interested in clinical neuroscience? I think I
Yeah, I think
can take that question for I can answer this question. So first, I think, the skills you learn in this competition will be very transferable to clinical research in neuroscience, or in fact, even research that uses a large scale data set in general. Okay. Secondly, I think fly neuroscience plays a crucial role in our understanding of many, many complex behaviors, but also how and why those behaviors goes wrong. And that is the fundamental way of researching of different neurological and psychiatric disorders. I also think that, the specific data set that you're also using also have many clinical implications. So as Arushi mentioned earlier, there are many behaviors that are conserved across species, from fruit flies to rats and humans. And And indeed, many complex behaviors are guided by vision. Or in other words, the visual system interacts with many other processes like reward, learning, and attention, and all of which are very, very essential for the species survival. And then when that piece of the jigsaw puzzle goes wrong, then there's all sorts of problems with your health and with your cognition. And of course, I think you will be very, very proud of the work you put into your research report and, of course, also the creative submission. And during the process, you will definitely come across studies in neurobiology that, or computational neuroscience, that could be both experimental and also computational. And being familiar with these studies can help you tailor your emails when you're applying to neuroscience research labs, since many of the underpinning behaviors rely on the neurons in the dataset that you'll be working with. So you sort of have the flexibility to explore like most of the behavior of interest and then research the neurobiological processes behind such behavior, but you're also leveraging the cutting edge connectomics tools. And I think that is why, like, by being a part of this competition, like, it would really help someone interested in neuroscience in general or interested in the medicine or clinical aspect of the brain. I hope that helps. Okay, how would you know if the neuron is at the surface or in depth? Everything looks the same to me. That's a great question.
That is a very, very fair point. So, on the surface or if in depth. So, anything that goes inside the neuropil, like this is inside the thing itself, that is inside it. Surface, you need to play around a bit. So, you see, this is another new, this is the lobula plate, this is the medulla, and it's sort of going at the top of it, like here, and it's going at the edge. But to characterize these neurons in a way, that's biologically meaningful for the purpose of the competition, I would suggest you see where it goes through. I know it looks really confusing when I started out using this. It's a very different sort of science. It's a very visual. You just have to practice looking at things and saying like, oh, this is actually going through. And when you read the literature, you'll actually gain and appreciate why we have this Neuroglancer, because you can see things in 3D and you will get a better understanding of what it means to go in depth or go through. So don't worry about it.
Could I participate in the competition even though my interest strongly lies on the research and technology side of neuroscience rather than medical or science fields? Yes, I think so. So in your research report, it's very flexible. We want to see that you cared about reading the stuff and you're very curious to explore. So if you are interested in, let's say, on the more computational side of things of the neurons, you could even look at some studies that you can basically read a lot of literature on the connectomics, on like image reconstruction, and you can incorporate it in the research report, and that's absolutely okay. And I think we want to make it as flexible as you can. And I think by doing that, you can demonstrate you're very curious about the brain, but also how did they reconstruct some of these, neural image and things like that. So as long as you show some insights into whatever you have read, that is perfect. Also, you could, it's a challenge, but it's not necessary for the competition. So actually these neuPrint, especially neuPrint, which is, a platform to analyze the connectomics dataset, you can query it using Python. Although not necessary for the competition, you can like use the web-based interface, but you can attempt to read the documentation, because if you're interested in programming, there's very good documentations that are available. And you could attempt to actually, try using simple Python queries to sort of interact with the data through computer science. But again, that is not necessary for the competition. But if you wish, you can. Hope that answers the question. For me, I did not have any background in neuroscience, but I do some computer-aided drug design, so I'm excited to do something new. Yeah, that's excellent. And I think eventually, neuroscience, pharmacology, neurological disorders, all of these are interconnected with one another. And, many of the pharmacological studies need really in-depth understanding of neurobiology, but also computational neuroscience. So I think you're on the right track. And I hope you have fun in the competition. So Celestine says, I still don't understand. I'm assuming the question related to the neurons at the surface or in depth. Before we go to Celestine's question, I want to ask another question. Who created these connectomic tools like neuPrint and Neuroglancer?
Yeah. So connectomics is a field where, like when we read about science when we're young, it's like this one person with a test tube, really going for it in their lab. But connectome is a field which is all about collaboration, all about working together. And we didn't build connectomes just by one single person, something. It was a massive collaborative effort. So neuPrint was built by HHMI Janelia. Um, Janelia is a research institution, and they are the same people where the optic lobe connectome, which you all will be playing with, comes from. And it's basically a database which stores many different connectomes, like the hemibrain connectome, the ventral nerve connectome, and all that sort of stuff. So they created neuPrint just for that. Also, keep in mind, connectome data are like, I think the full adult female fibrine is like 100 terabytes in size with like 20 million images or something. That's massive. You're gonna crash your computer. I don't even think your computer can store that data. So this is why these ecosystems exist. So we can query exactly what we want and what we need out of it. Neuroglancer was made by Google Research. So again, these connectomes were made in collaboration with HHMI, Janelia, MRC at Cambridge, Princeton, and Google Connectomics, and there are other institutions that are escaping my memory, but it was a large collaborative effort, and Google Research came in and has provided many tools like a lot of AI tools and also stuff like Neuroglancer to actually visualize this connectome data.
Yes, and thanks to them, we now have something that is so accessible to all of us, which is pretty cool. Yeah, great. So let's see if we had any more questions. Of course. Yeah, I'm glad that Connectomics, you're quite interested in it. And, feel free to ask many questions. We are here to help. And, we are very active on Discord.
Mm-hmm.
And join our Discord. You can ask on any channel. We have like Ask the Staff channel, or if you want to ask in the Fly Connectome channel, that helps. And we will try and we'll try our best to be very responsive. But Arishi, can you go ahead and maybe
Mm-hmm.
clarify, since we have time, 7 more minutes? Yes, maybe, yeah, choose another neuron. That would really help. And I think, visualization is one of the coolest thing to admire the beauty and the complexity of the brain, and it allows us to make many hypotheses about neurons. So I think these questions are really worth it. So thank you so much for asking them.
Okay, I think this is a good one. Okay,
Great.
so remember ROI is on. This is what's going to help you see if it's inside a neuropill, which is these four lamina, medulla, lobula plate. So you need that on to see. This neuron here, this is T5A. It is inside the lobula, you can tell. But here, you can tell it's in between the chiasm. It's in between like nothing, no neuropil here. But now it's also in the lobula plate, this part with the cell body. So that's how you know it's inside something or if it's not inside something. Maybe like another example. Okay, so this one, this is a neuron, this is a visual projection neuron. It goes to the brain. So you see it's kind of going outside. For the purposes of this competition, it's optic lobe and it's okay. But remember, the point is to try your best and characterize. Remember, there are anatomists out. If this connector was, it's not easy to characterize these things. There are experts out there who know the fly brain more than the fly brain does. So there, it's not, don't worry, don't feel scared. It's not easy. Try your best to characterize this. There are people who sit there and literally look at things and see if it's a cell type or not. Not easy. So as you can see from this LC10A, this is in the lobula, and you have to rotate things around a bit to see. So you can see this is in the lobula, and it's going to the central brain, but over here. But you see the central brain is not annotated over here because this is just the optic lobe connectome. So you can see it's going in there. But yeah, so this is all the lobula. Yeah, just play around, and I'm sure it will make sense. And remember, you try your best. Knowing which layers it's going in is a lot more important than which one it's just surface passing, because which layers it goes in is telling you more about what it's doing. Like if a neuron is in the medulla, that is an important thing to know, rather than it's kind of escaping the lobe. So something like that.
Perhaps if you could show an example neuron again and maybe point where
To
is the
this,
soma and where's the dendrites? Because I think it's a bit hard to
yeah, yeah, no, absolutely, absolutely. Um, okay, I zoomed in too much. So
Great questions, guy.
yeah, thank you so much. Thank you so much for it's helpful for me to know things as well, like what is not clear. So, okay, remember what I said about the fly. It is literally really hard to tell where the axon and dendrites are just by your eyes itself, unless you're a neuroanatomy expert, I imagine. So they're kind of all over the place, but the soma is very obvious to tell. It looks like a little blob. So this is the soma right here. And this is a combination of, I don't know what it is, axons and dendrites. So the presynapses, you see the presynapses where it's getting input connections from, they're on both of these arbors over here. And the post synapse, where it's sending outputs to, is also on both. So it's really hard to tell which one is the pre and the post synapse. So that's really hard to tell. So, but you can tell that's the soma. But the axon and dendrites are really hard to tell, just like that.
Can you repeat that? So actually, the pre-synapse and post-synapse. Oh, that's
So no, they're both. They're even here and they're also here. Like it's not that's the fly neuron is kind of a mess. It's not very it doesn't have clear polarity. The axons and dendrites are intermeshed together as you can see here. So that's what I'm trying to say, right? Like even I can't exactly tell like which is the post and pre. It's just hard to tell unless you're an expert neuroanatomist. So just to know, but these are the axon and dendrites, I would say, and this is the soma over here, the cell body.
I think the rule of thumb is that you could use Neuroglancer and click the eye on the pre-synapse and post-synapse just to see where things are, and then you will know for that individual neurons. In terms of NeuPrint, so you just use the NeuPrint to know the pre and post synapse
Yeah, you can see the pre and post synapse connections in
NeuPrint.
neuPrint. Yeah, yeah.
And I think NeuPrint just gives more details as to which specific neurons are inputting into that neuron cell
Hmm.
you're interested in, and whether that neuron cells outputs to which other neurons. And of course, it shows
Hmm.
like the top five connections and so on. So I think that's where NeuPrint really
So
helps.
neuPrint, yeah, helps you tell what it's connecting to. Because in this, it's gonna be it's this is why it's not easy by eye exactly. And these these reconstructions happen from these EM data sets here. So you see, you actually have to use these and people out there like see what it's connecting to from these are all like individual neurons and stuff out here. So that's why for the purpose of honestly like most people even like me, this is to help visualize the structure of the neuron, like okay, where is it going, and just to get an idea of oh, okay, this is what it looks like. Whereas Newprint can help you see like what it's connecting to and that sort of stuff.
Great. And also keep in mind, although these data sets are very accessible and the concept itself is visualization and then seeing the connections, in reality, it's often you might need to, play around with the data and or with the platforms. These things do require some intuition, but it is now made to the point where the learning curve hopefully is not as steep as it used to be. Yeah, I get you. Like, there's a lot of reading to do, but then again, right, that is the point of the competition. We hope that this inspires you now to go read something and actually have a sort of a goal and a sense of purpose to kind of read. Before it might be like you're reading aimlessly, but now there's a goal, to actually use these tools to come up with new insights into the neurons or even learn the technical sides of the connectomics, which is essentially computational neuroscience and also relevant to experimental neuroscience and so on. Yeah, so I really hope this is helpful. Thank you again, and we hope you have a great weekend.
Thank you. Such a pleasure. Thank you for all the incredible questions. I also learn a lot. So thank you.
Okay, thank you very much. Bye bye.
Thank you so much. Thanks, Aponax. Thanks, Arushi.
Thank you.
Clematis Research Empowerment Hub. Empowering research for everyone.